Welcome back, folks, to section 6.8 on our AP Bio unit on genetic um, control. And this one, we're going to do a little bit of an introduction on some techniques in biotechnology about how we can manipulate this stuff. This is not a biotechnology class. We're not going to be learning all the details, but just to give you a general overview of some of the major things that we might see on the test. So, biotechnology in general is the manipulation of organisms or their components to make useful products. Okay. Biotechnology. Genetic engineering is the direct manipulation of genes of those organisms for practical purposes. DNA sequencing, very simply, it's a technique of determining the sequence of nucleotides in DNA. So, it's figuring out, okay, for this organism or that chromosome, what order do the A's and T's and G's and C's come in? We've talked about it before, but just to remember that we have DNA ligase, which is an enzyme that repairs breaks in DNA, join DNA together. In this instance, we use it to make recombinant DNA, which we'll talk about a little bit later. Um, so, looking at that, we can take a bacterial plasmid, a gene that we want to make, say insulin, a human insulin gene. We can put it in the plasmid. We now have a recombinant bacteria. It's got a combination of the original bacteria genes plus the gene we want to make more of or get into something, and then we can use that to uh, get plants that are resistant to pests, bacteria that can help clean up toxic waste, human growth hormone, or just make bacteria that make human insulin directly. It's the exact human insulin protein that we can use in our insulin today. Gel electrophoresis. Um, for this one, guys, what do we use it for? And what should you pay attention to? What I've done here, guys, is there's an Amoeba Sisters video on this that I really like, that I think does a really nice job of going through it with visuals that I think are going to be very helpful for you from what I've seen from all the questions I'm able to gather on the AP Bio exam um, from the past and everything else. And I don't think I can do a better job. So these are the things I want you to look at while you're watching that video and maybe jotting some notes down on. Um, and then we'll come back after the video to watch it. So I want you to take a look at, we're using it mainly to compare differences and similarities between DNA. That's the big picture. I want you to pay attention to how does DNA size affect its speed and distance traveled. That'll make more sense when you watch the video. What are restriction enzymes and why are they important? And I want you to note and remember that DNA is negatively charged, which causes it to be attracted to a positive charge on the gel. So let's watch that Amoeba Sisters video. We'll come back afterwards, highlight a couple of things, and then move on.
When I was a young kid, I was really interested in genetics. Well, I didn't really understand genetics. I kind of thought that when two organisms had a baby, the baby was just this blend of the two. Yeah, that's a misconception. But I really saw genetics in action with my guppies. Guppies are very easy freshwater fish to keep in an aquarium. But they have two things that I think are especially cool. They have live birth, which means there are no eggs, like many other fish. And second, they have a lot of babies. They also eat their babies, but I don't think that's especially cool. So as you can see, that is not part of my cool fact list. Anyway, when my surviving baby guppies grew up, they would have all kinds of cool traits. These traits were carried by their DNA, their genetic material, which is found in their body cells. But sometimes I would forget which mother was the mother of the baby guppy, because there were several mother fish in the tank, and I wanted to keep track of inheritance in my guppy notebook. So what would have been very cool to have at that time? Some biotechnology. Biotechnology is the merge of biology and tech, and it's constantly changing. It includes topics such as PCR, cloning, and genetic engineering. It's also an awesome field. We're going to talk about one of the biotechnologies that could have, well, potentially, helped me determine the genetic relationships of my guppies. If, as a young kid I had access to it, although it's becoming way more common in classrooms now. And that biotechnology is gel electrophoresis. Gel electrophoresis can be used to separate molecules based on how big they are, their size, and it's especially useful with DNA. Let's look at DNA real quick. So here is a guppy cell, here's the nucleus in the guppy cell, here's the DNA in the nucleus of the guppy cell, and if you were to zoom into the DNA, here is a nucleotide, which is a building block of DNA. See those phosphates in the nucleotides? They're a bit negative. Well, they contribute a negative charge anyway to the DNA. So if we look at this whole DNA here, it gives that DNA a negative charge. That's a big deal because gel electrophoresis, which again separates molecules based on size, relies on the fact that DNA molecules have a negative charge. Okay, here's a gel electrophoresis machine. The point of the machine is to be able to have an electrical charge running through a gel. So here's the gel, typically made of agarose. Agarose is a polysaccharide polymer, which, if you remember from our biomolecule video, polysaccharides are carbohydrates. Yeah, usually agarose comes from seaweed. The agarose gel itself lets the DNA molecules travel within it. One end of the gel has these holes called wells. The wells are where the DNA is placed into. The area of the gel where the wells are is negatively charged, and the area of the gel here is positively charged. So guess where the DNA will travel towards? Well, since it's negatively charged, it's going to travel to the positive side. So typically when you're analyzing DNA in electrophoresis, you use these restriction enzymes to cut the DNA up into tiny pieces. Restriction enzymes have the ability to cut up DNA in very specific areas, often related to the specific DNA bases, making restriction enzymes very useful in biotechnology. So if I had baby guppy DNA and adult mother guppy DNA, and I want to compare them, then I would want to use the same types of restriction enzymes in both DNA samples. If I used the same type of restriction enzyme, it should be cutting the DNA at the same identification points in the DNA samples. However, unless the mother and baby guppy are clones, and they're not, those pieces that result after the restriction enzyme is done with them may be differently sized because the DNA of the baby and mother guppy had some differences in the sequence of their DNA bases. So the DNA samples both are cut up into multiple pieces by the same type of restriction enzyme, and then those samples are loaded into the gel. Sample 1 and sample 2. If we turn on the machine and let the DNA run through the gel, the DNA moves towards the positive side. But some pieces of that cut up DNA will move faster or slower than other pieces. Longer DNA pieces tend to have a higher molecular weight. They take more time to make it across the gel when you compare it to shorter DNA pieces, which move at a faster rate. So what you end up with is that these DNA fragments spread out, with the longer pieces closer to the wells and the shorter pieces closer to this opposite side of the gel. These are called DNA bands, but to see them, you usually need to stain the gel itself and view it under a UV light. Now let's compare the DNA bands in this hypothetical, simplified guppy situation. The bands aren't going to be identical because these fish are not clones, but I can compare how similar the bands are and compare that to other mother guppy samples to look for relationships. Let's say that we have 3 mother guppy samples to view, and these are the only possible mothers from the fish tank. Which one appears to be the most related to the offspring in this case, and has a high likelihood of being the mother? Well, this one. But we can't be 100% sure with this. It would be helpful for me to know the father guppy sample too, because this will give me more insight. But if these are the only fish in the tank, it's a very high likelihood with this case. Also, you can use something called a DNA ladder. You can buy them from various science material distributors, but a DNA ladder is not what it sounds like. It's basically a sample that has known fragment sizes. So if you run it in the electrophoresis machine, you already know the fragment lengths. Let's say this DNA ladder only had 3 bands, which is not usually the case. Since it's a DNA ladder, the base pair lengths are known. They are 500 base pairs, 1,000 base pairs, and 1,500 base pairs. Think for a minute. Where would they fit in? It would look like this. You can use this now as a reference to give estimates of how large the other fragments are when they're run alongside it. And if you want to be closer to the value, you can use a semi-log graph, something to look up. So why do we care about gel electrophoresis? It's not likely I'll be actually using this with my guppies anyway, right? Well, perhaps, but gel electrophoresis is often a step used in determining relatedness with different species, which helps scientists better classify organisms. It's also used as part of DNA fingerprinting. DNA fingerprinting is a way that one can identify someone's DNA, which can be very helpful if you're trying to solve a mystery involving a crime scene. If you have a DNA sample from a crime scene, you can go through the steps of gel electrophoresis to compare it to the suspect DNA to see the likelihood of a match. In fact, you can take the results from gel electrophoresis and isolate genes of interest by something called southern blotting. Definitely something to look up if you're curious. Gel electrophoresis is one of many awesome tools in biotechnology. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So, just a little bit more about restriction enzymes, which we use to make recombinant DNA. There are restriction enzymes which come from bacteria, and their job is to cut DNA molecules at specific DNA sequences, like AATAA, for instance, might be a restriction site. Um, and these restriction enzymes usually make many cuts because any given strand of DNA, there's going to be a lot of places where you get AATAA, for instance. So it'll cut it there wherever it finds that. So it cuts them into random, well, not randomly, it cuts them at those sites, which winds up making randomly linked chunks of DNA. Um, the most useful restriction enzymes actually cut DNA in a staggered way, producing fragments with what we call sticky ends, which we combine with complementary sticky ends of other fragments. And we use that as part of the way how we make recombinant DNA. We can break apart some DNA where we want it, and then we can stick it onto other DNA because of those sticky ends, and then DNA ligase can come in and bind it all together into one piece. So let's take a look at that. So here's our bacterial plasmid, for instance, with a restriction site. In this case, it's GAA-TTC, and the complementary site over here. Restriction enzyme cuts it, but staggered like this. So we've got these ends here that aren't bound to anything, so they really want to bind to something with the complementary end, this over here. So we can take that and we can grab a piece that we cut with the same restriction enzymes, because the same restriction enzymes will leave the same sticky ends. And then this piece can now fit between these two pieces, and we can insert that new bit of DNA between the other bits of DNA to make our recombinant DNA molecule. And then we can use that for all the things we can use recombinant DNA for. And here's a little video that shows that. We'll catch you on the other side.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAA-TTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
PCR, or polymerase chain reaction, has been in the news a lot the last couple of years, so you're probably already familiar with it, at least in general, but we'll make sure there's at least a clear definition here. It is for amplifying DNA or making more of a particular chunk of DNA. So the polymerase chain reaction, or PCR, can produce many copies of a specific target segment of DNA. So we want more of this part of it. And the reason we may want to do that is so we have enough of it to work with, enough of it to do DNA sequencing or to do gel electrophoresis or whatever it is we want to do with that DNA. From any one sample, you're not going to get a lot of DNA. So by using a PCR amplification process, we can make from a little bit, make a ton. Okay? And it's a 3-step cycle. First, you heat it up, which we call denature it, because we know heat can denature things. Cooling, which the technical term is annealing, and then replication, which is where we're elongating it. We're actually replicating that chunk of DNA. And it brings about that chain reaction that produces an exponentially growing population of identical DNA molecules. Okay, so the key to PCR is an unusual heat stable DNA polymerase because we need a DNA polymerase that can handle those high temperatures that we use for denaturing, and that's called Taq polymerase. Um, and we also use a pair of primers. Remember, we have RNA primers, right? Specific to that sequence that we want to amplify, so we amplify the right part. And for instance, this is how we're amplifying the DNA to do a COVID PCR test. So you can start off with your target sequence here, little chunk right here in the DNA. We denature it, which basically unzips the DNA, anneal it. When we cool it, we get those primers attached. Okay, and then extension or replication. That's when we actually build the other half of it, and now we got two new chunks of that DNA of the original up here. Now in the next cycle, we double that. In the next cycle, we double it again, and so we get exponential growth relatively quickly that we can basically force replication to happen on certain areas of the DNA that we want to work with. Lastly here, guys, bacterial transformation. We did mention this before, so if you recall, a transformation is when a prokaryotic cell can take up and incorporate foreign DNA from its surrounding environment in a process called transformation. So we grab naked DNA here in a plasmid form, suck it in, and now we've got a bacteria that has DNA from itself and from somewhere else. Why is that useful? Well, we can add genes and thus phenotypes to new bacteria that never had them before. We can use them to make proteins like insulin, we mentioned before, to do gene therapy, where we can insert genes into people and get them to make their own insulin, for instance, is one thing people are hoping to do, and lots of other things. And that's all the deeper we're going to go into this stuff, guys. I think that is all we need for the AP Bio exam as far as biotechnology goes. From the research I've been doing, it looks like you're most likely to see stuff where you actually have to interpret a little bit deeper the stuff on gel electrophoresis, which is why I put the Amoeba Sisters video in there for you. So if you got questions, ask me in class, and we'll catch you next time, guys. Bye.